Let's take a closer look at the structure of a nephron, starting with the renal corpuscle. As mentioned previously, the renal corpuscle has three parts. The outer capsule, called the glomerular capsule or Bowman's capsule. Then there's a space called the capsular space, which collects the filtered stuff that comes out of the blood, and we call this filtered stuff filtrate. And then there's the glomerulus, which is a fenestrated capillary that's inside the corpuscle. The glomerulus is a fenestrated capillary, and it's coated with cells called podocytes. And we'll talk more about these fenestrations and podocytes in just a minute. But remember, the blood that's being delivered to the glomerulus comes in through the afferent arteriole, and then once the blood is in the glomerulus, we'll squeeze some substances out of the blood, pushing them into the capsular space to form the filtrate. But there's still stuff left in the glomerular capillary. We need that substance to leave the glomerulus, and it does so through the afferent arteriole. The function of the glomerulus is filtration of blood, and this filter works very much like a coffee filter. It's size selective. Substances that are too big can't get through the filter. The filter here is going to be composed of really two things: the fenestrations in the wall of the glomerular capillary, and then also things called filtration slits that are formed by cells called podocytes. So we're already familiar with how capillaries work. Substances can move out of the capillary by crossing the capillary wall by going through these fenestrations. It turns out that the outside of the glomerular capillary is coated with a type of cell called a podocyte. The podocytes, though, have little spaces between them, and those are called filtration slits. And like a fenestration, they are size selective. So really small things can get through, but bigger things like proteins can't get through. So in order for the filtrate to form, the substance has to cross the fenestrations of the capillary, and then they have to go through the filtration slits that are between the adjacent podocytes. At that point now, the substance will be in the capsular space, and we will call it filtrate. So we've said that things like cells and proteins are too big to get through. So what sorts of substances will get through and will be found in the filtrate? Well, there'll be a lot of water. There will also be a lot of metabolic wastes that are produced when we use things like proteins for energy, like urea and ammonia and uric acid. Creatine is another one. We will also find ions, or electrolytes, if you will, in there, like sodium and potassium. Toxins and drugs also can make it through those fenestrations and filtration slits if they're small enough. What I'd like you to do right here is to pause the slide and answer some questions, and once you think you have the answers, go ahead and hit play. So the questions are to name the spaces shown in the image. So we just finished talking about the renal corpuscle and what's happening in there. So keep that in mind and apply that to this situation. So you're going to name a couple different spaces, and then you're going to identify the things that are noted by the arrows. So go ahead, pause the video. When you're ready, hit play to see if you got the answers right. All right, let's see how you did. So what do we know? Well, we know these orange squares are the podocytes, and that's all we're really given here. All right, so we know that we have spaces between adjacent podocytes that are called filtration slits. So this area, or this arrow rather, has to be denoting the filtration slit. So this over here looks like it's got holes in it too. So this is our fenestrated capillary. And these holes are the fenestrations in the wall of the capillary. So this space is going to be the lumen of the glomerular capillary. The thing that's denoted by this arrow here would be the cell wall of the glomerular capillary, which is going to be composed of an endothelial cell. Last, we have the space shown in green. This will be the capsular space that will be collecting the filtrate. Let's do this again with this image. So you're going to pause the video and then hit play when you're ready. What questions are you answering? In this case, I want you to identify the structures that have been circled in black, and also to identify some things and a function for the area shown in the black rectangle. Now keep in mind, again, we just finished talking about a renal corpuscle. So keep that in mind as you answer these questions. All right, let's see how you did. So A is referring to the whole entire corpuscle, right? So the A is referring to our renal corpuscle. Now we know the majority of what's inside our corpuscle should be the glomerulus, so number 10 is representing our glomerulus. And we can see that we have a vessel leading into the glomerulus and then leading out. So number 9 is the vessel leading into the glomerulus, so that has to be the afferent arteriole. And number 11 is the vessel leading out of the glomerulus, so that would have to be our efferent arteriole. We also have to label number 4, which is the space of the renal corpuscle, so that makes that the capsular space. Number 3 is a little bit more difficult, but if you recall, the outside of the glomerulus is coated by podocytes. So number 3 is referring to the podocytes. B is representing the proximal convoluted tubule that will be pulling the filtrate out of the renal corpuscle, bringing it into the nephron's renal tubule. The last thing we have to talk about is the area in the little box. So you can see that area is denoting the connection point between the wall of the glomerular capillary and then the podocytes. So you can think of this as like the filtration membrane. This is where the actual filtration process will occur, where substances have to cross through those fenestrations and then through the filtration slits in order to make it into the filtrate.